There are two mechanisms how ATP could be generated during breakdown or catabolism of glucose. So one mechanism is called substrate level phosphorylation. And during this process, enzymes have two active sites. One for the substrate which have phosphate group and the other for ADP. Then enzyme will change conformation, shape, and it will bring phosphate group close to ADP molecule. A new chemical bond will be formed between phosphate group and ADP. And bond between substrate and phosphate group will be broken. And result of this will be ATP molecule will be product because when we're adding phosphate group to ADP, we're creating three phosphate groups, so ATP. And substrate now will be completely different molecule because we remove phosphate group, so this will be product. And in here showed you as example of this substrate level phosphorylation. We can produce ATP when we have enzyme with two active sites, one for ADP, 2-phosphate group, the other for substrate, which have phosphate group. Then enzyme will change shape. Phosphate group from substrate will be detached and attached to the phosphate group in the ADP molecule, and ATP will be released, and without phosphate group, substrate become product, will be released. The oxidative phosphorylation is the second mechanism how cells can produce ATP during glucose catabolism. And chemiosmosis is a process which used in mitochondria of the eukaryotic cells or on plasma membrane of prokaryotic cells, for example, bacteria. Chemiosmosis is a process of production of the ATP molecule during cellular respiration, and it generates 90% of the ATP during glucose catabolism. Chemiosmosis is also used in the light reaction of photosynthesis to harness, to collect energy of the sun. And in here show image of mitochondria, and this is outer membrane, and inside is the inner membrane. Glycolysis is the first step of aerobic respiration. Glycolysis happens in almost all cells of the all living organism, and it's an anaerobic process. It means it doesn't not require oxygen. An mean negative, no aero mean oxygen. Glycolysis occurs in cytoplasm in both prokaryotic and eukaryotic cells. Glycolysis converts glucose, which is ring-shaped molecule and have 6 carbons, into the pyruvate, 2 pyruvates, which contain only 3 carbon molecules. And energy first required on the first step of the glycolysis in order to break down bonds between carbons, but later on in the chemical reaction, not H will be produced. So whereas H means it's carrying energy, electrons, and plus 4 ATP molecules will be produced. But if we calculate total net production during this glycolysis will be from one glucose molecule, we produce 2 pyruvates, 2 ATPs, because 2 will be used in here, and 2 not H molecules. Oxidation of pyruvates will prepare pyruvate to enter citric acid cycle. After glycolysis, next step is a citric acid cycle, but pyruvate cannot directly enter this cycle, so pyruvate need to be converted. And it's happening when pyruvate in here first in the cytosol is produced. This is show part of the mitochondria, outer membrane, inner membrane with matrix. So pyruvate first will be moved through the outer membrane, then it will use special transmembrane protein called pyruvate carrier protein to move pyruvates uh, through this inner membrane inside of the mitochondria inside, and in here will be conversion. So pyruvate will be break down with help of the coenzyme A to acetyl-CoA. And during this conversion, what happened? CO2 will be produced and NAD will be reduced, become NADH, and will carry energy. Citric acid cycle is the second step of aerobic respiration. And during this cycle, acetyl-CoA will be converted into the carbon dioxide. And it will happening only when oxygen present, of course. And during the citric acid cycle will be continuous harvesting, collecting this energy from the remains of the glucose molecule, and in the end will be released into the oxygen. Carbon dioxide. So during the citric acid cycle, acetyl-CoA molecule will be converted into carbon dioxide. And this cycle happening in the matrix of the mitochondria. And in the end, from one acetyl-CoA will be produced two carbon dioxide molecule, one ATP or GTP, it's similar, and NAD will be reduced to NADH and FADH. So they both have H, so they both carrying electrons energy. Citric acid is aerobic pathway, and citric acid cycle have alternative name, Krebs cycle, because of the name of the scientist, his last name Krebs, who first identify and discover these steps. And on this image, you can see how acetyl-CoA entering first this cycle. It's called cycle because how you can see one converted into the other. And during this cycle of the chemical reaction, water will be used to break down chemical bonds, and NADH will be produced, and carbon dioxide will be gas produced. And in here more carbon dioxide and no NADH is produced. And in here ATP is produced. And in here FADH is produced. In here water is used. And in here again NADH is produced. So this is 8 steps happening all the time. Oxidative phosphorylation is the third step of aerobic respiration. And first we will start with part A, electron transport chain. Electron transport chain, or ETC, include step. So this is the step of the aerobic respiration, and it's only one which directly use oxygen. You can see in here oxygen we have. And it's called like this, electron transport chain, because you can see this is different proteins. and they will perform reactions, chemical reactions. And during this reaction, electrons in here E, this is electrons, will be transferred from one chemical reaction to next, and in the end, they will be giving to oxygen. These electrons in the end will be attached to oxygen in form of the hydrogen, and water will be produced. So electron transfer chain is a set of the several transmembrane proteins, and these proteins called electron transporter proteins because they transport electron. So they will use in here our electron carriers such as NADH and convert it to the NAD, and H will be moved from NADH. Protons will be moving from this is inner mitochondrial membrane in here, and they will be moving in the intermembrane space in here. This is mitochondrial matrix. So NAD H located in the matrix. Then this protein, first electron transport protein, will transport hydrogen and protons, H plus is protons, through it across intermembrane into the intermembrane space. And during this, electrons will be moved to the next protein collected in here, and hydrogen protons will be in this intermediate space. So then next reaction happened. The other electron transporter will be moved from the fat H, moving electrons, and converted fat H to the fat. And electrons will be moving to the next electron transporter. And during this reaction, again, hydrogen will be moving, transported inside, and electrons will be passing to the other transporters. And in the end, oxygen will be binds with electrons and produce water in here, and protons will be accumulating in the intermembrane space. So we're pumping kind of hydrogen ions through these electron transporters and collecting electrons and producing water from oxygen. So what is important, remember that function of this electron transport chain to establish hydrogen ion gradient. We making on different side of this mitochondrial membrane, we're creating gradient. We have a lot of hydrogen protons on one side, and in the mitochondrial matrix, we have less. So a lot of positively charged protons accumulating in here. So now we will talk about part B of the oxidative phosphorylation. It's called chemoosmosis. And during chemoosmosis, this is the last step of the oxidative phosphorylation, will be produce a lot of energy. And we will have transmembrane protein enzyme in here called ATP synthetase. A is telling that it's enzyme, and ATP synthetase telling us what it will be doing. It will be synthesizing ATP. From ADP and phosphate will be produce ATP. But we need a lot of energy to connect phosphate group to the ADP to produce ATP. So where is this energy will be coming from? And energy for chemoosmosis will be coming from the concentration gradient of the protons. Remember, we're accumulating a lot of hydrogen protons in the intermembrane space. and we have low concentration of the hydrogen protons in the mitochondrial matrix. So when this channel will be open, hydrogen protons will rush from area of the high concentration to area of the low concentration, and this will be energy, and this is what will help to attach phosphate group to the ADP. But hydrogen ions not only creating concentration gradient, they creating also electrical gradient. Electrical gradient means that on different side of the membrane we have a lot of one type of the ions, in our case hydrogen ions, which are positively charged. And on the other side of the membrane in here, where is mitochondrial matrix, we will have opposite negatively charged because we was pumping these hydrogen ions. So we creating here negative charge on this side of the membrane, and we creating a lot of positive charge with these hydrogen protons on the other side. So hydrogen ions not only want to move from area of the high concentration to area of the low concentration of them, but also because they positively charge. And inside in here on the other side is negative charge, and negative charge is attracted. So positively charged protons will also rush inside of the mitochondrial matrix because of their charge. They are positive and in here negative charge. And purpose of this chemoosmosis is to produce ATP. A lot of ATP will be produced in here. So in here we have common kind of overall oxidative phosphorylation, which combine two processes, electron transport chain in here on this side and chemoosmosis on the other side. Remember, during electron transport chain electron transporters, these proteins, will be transporting electrons from the node H into in the end to oxygen and will convert it to the water. In the same time, they will be pumping on opposite side of the intermembrane in here. In the membranous space, they will be moving hydrogen ions. So they are accumulating in here. And this create gradient, both concentration, high concentration of the hydrogen ions and low concentration of the hydrogen ions. And electrical gradient, we have a lot of positively charged ions on one side, and overall in here we're creating negative charge. So hydrogen ions in the last step of the oxidative phosphorylation chemoosmosis will rush inside of the mitochondrial matrix because they're moving from area of the high concentration to area of the low concentration, and because they want to move to the area where is opposite charge. In here we have negative charge and hydrogen protons, they are positively charged. And all this energy from the movements of the hydrogen ions will be used by the ATP synthetase to attach phosphate group to the ADP and produce ATP molecule.